This is TWIS. This Week in Science, episode number 798, recorded on Wednesday, November 4th, 2020. Science is all about patience. Hey everyone, I'm Dr. Kiki, and tonight on the show we will fill your head with glowing animals, little nudges, and earwax. But first, thanks to the Burroughs Welcome Fund and our Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer, disclaimer, disclaimer. Math! You either have it on your side or you don't. Whatever numbers you are working with, it's never going to add up to anything other than the sum of its parts. You can wishful think, you can squint your eyes, look sideways at the equation, and if you like, you can stand on your head to get a different perspective. But math has a way of being perniciously indifferent to how you approach the solution. And as ballot counting continues in America, millions await the fate of a nation, now at the mirthless mercy of math. But don't blame the math. After all, without math, we couldn't bring you This Week in Science. Coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back, like I said we would be.
Is there a chicken in here? Bark, bark, bark. Bark, bark, bark.
No. Bark. Cock-a-doodle-doo. Okay, I'm not a chicken. I can talk about science. It's gonna happen. We're gonna talk about all the science. We are in a mood tonight, and I hope you are in a mood to listen. So, on tonight's show, what did I bring? I've got some earwax, as mentioned in the very start. I've got some missing height as well, and I've also got a few nudges for you. We're going to talk about nudges.
Oh.
Those things that maybe you don't really know are happening. Nudge, nudge. Justin, what did you bring?
I've got early human lady hunters, invasive rats, a leukemia super spreader, ancient Asian genomes, and why magic mushrooms might be a magic bullet in the treatment of depression.
Sounds
Sounded
amazing.
a little too news anchory, actually. But
Very news anchory, maybe a little salesy, but we'll talk about that when it comes up. Blair, Animal Corner, please.
Yes. I have biofluorescent platypus. I have dull-colored birds. And I have surfing fish.
Surfing. If I were a fish, I'd want to surf. Why not?
Yeah.
Okay,
But do you surf on whales? Because that's actually what this is about.
This is fish extreme sports.
Yeah.
No, extreme ichthya sports. All right, let's dig into the show. As we jump on into all these stories, I would like to remind you that if you are not yet subscribed to This Week in Science, you can find us anywhere podcasts are found. Stitcher, Spreaker, Spotify, Pandora, radio.com, tune in, all over the place. Apple, Google. You can find us also on Facebook and on YouTube and on Twitch. Look for This Week in Science.
are those all real?
Yes.
Wow.
People have so many options these days.
That's
it's amazing.
wild.
There's also
Just almost any one-syllable word, it turns out, is a web client. So
it is
out there, especially if there's lots of consonants.
Consonants like calendars. And I want to remind you as well that calendars are available. We'll remind you again later in the show. But yes, Blair's calendars are for sale on the TWIS website. Head to twis.org. Click on that froggy toad. And you can buy a calendar.
Mhm.
Yes, let's plan ahead for another amazing year. Okay,
no, just an amazing year, but yes.
and an amazing year.
Yeah.
So let's talk very briefly. Let's move into the news. Shoo-choo, short stories. Here we go. Short-ish, kind of. Speaking of missing things, not missing time, but maybe missing explanations for how our genes influence height. Since about the late 2000s, when researchers started looking into the genes of the genome to figure out what markers were linked to various diseases, and starting through twin studies and family studies and looking at the influence of wealth, they started picking out genes that are related to height as opposed to environmental factors like disease state in childhood or how good of food you got or other environmental factors. So the markers that they were looking at, they couldn't tally it up. They couldn't figure out of the DNA markers what explained all the height. They could only at first explain about 5% of its variation. And then they ended up putting together a number more genetic markers, and they got it up to about 40 or 50% of the genetic component of height. So they're getting there. But now, in about 2018, a team of researchers put together an effort called GIANT. It's a global consortium called GIANT, and it pooled the data of 700,000 people looking across 3,300 markers that then explained 25%. They added things up and added things up. But they have been able to put together a bunch of things, and basically they are tracking down and they're starting to put it together. They've identified nearly 10,000 DNA markers now that explain the influence.
And that's why it's so varied and why
Yes.
you're a mixture of both of your parents, but also it's not a perfect mix. And
why you can't say by just measuring a child and they're measuring the parents that the child will be 6'2" when they are 19 years old. There are a lot of things that go into it. We've been measuring this for a very long time. And even knowing all of these 10,000 markers that are involved, being able to use them like sliders on a mixing board to produce the height of a child is still beyond our abilities. But we're figuring things out. We're figuring things out. Yes. So we're getting there. We still have a ways to go. How tall will I be when I grow up? I don't know. I will probably be shorter because I'm already shrinking. Okay, speaking of women, Justin, tell me about lady hunters of the past.
Yes. So men hunt, women gather. When early human groups sought food, the division of labor was distinctly dictated by an individual's gender and how they participated within the group. That's just the way it was, or so somebody must have assumed and then convinced everybody else, so that became a saying, most likely reflecting the imaginary reality created by men with big sideburns, bushy eyebrows, and extraordinary nose hair mustaches. Because according to the research conducted at the University of California, Davis,
Go Davis!
It was absolutely nonsense. 9,000-year-old female burial in the Andes Mountains in South America reveals her to be a hunter.
Ooh.
Not a gatherer. She's a hunter.
Yes.
Randy Haas, assistant professor of anthropology, is the lead author of the study, which is titled Female Hunters of the Early Americas, which is published in Science Advances. Quoting Haas here. We believe that these findings are particularly timely in light of contemporary conversations surrounding gendered labor practices and inequality. Labor practices among recent hunter-gatherer societies are highly gendered, which might lead some to believe that sexist inequality in things like pay or rank are somehow natural. But it is now clear that sexual division of labor was fundamentally different, likely more equitable, in our species' deep hunter-gatherer past. So, This comes from one individual that they found, but it was sort of it posed a question. Hey, have we really looked at identifying who in this ancient past was a hunter? So they went back and they looked through all sorts of other individuals, 429 individuals from 107 sites. Of those, 27 were associated with big game hunting tools. So this isn't like hunting squirrels. This is like going after big prey. Of that 27, 15 were male, 11 were not. In fact, or 12 were not, I guess, but 11, or one maybe, there's one missing. I don't know what happened to it. 11 were female. The sample was sufficient to warrant the conclusion that female participation in early big game hunting was likely non-trivial. This 9,000-year-old specimen wasn't just a badass that was participating in the hunting, but it was more of a norm in
more common
ancient times. Yeah. So their statistical analysis shows that somewhere between 30 and 50% of hunters in these populations were huntresses. And even actually in the more recent modern human hunter activities, it can get up to 30%. So it's not completely removed. So anyway, change your views of how you think the division of labor naturally occurs in societies. Because I think, like, we've talked about this in Neanderthal studies. Like, everybody goes on the hunt, except for, like, the wounded that they care for that stay back at the cave or whatever. But everybody goes out and has the broken bones and the injuries associated with attacking the prey.
Whoever's capable, right? It's if you are able-bodied and capable, why not? And I think it would make more sense, I mean, especially in early hunter-gatherer civilizations, for anyone who's young and able-bodied to be on the hunter side of things, the gatherers to be the, you're getting a little bit frail, you're complaining about your back and your knees a little bit more. So it's like, Grandma, why don't you stay back at camp, or why don't you go look for the wild radishes?
Now, wait a second. Now, let's not. The gathering required a lot of stooping, a lot of
absolutely
up and down, picking things, carrying things like that. It was not easy work either.
None of it is easy work. I'm not. None of it. None of it's easy work.
So Thunder Beaver in the chat room is bringing up a really good point too, which is that we know that in Native American cultures, in many of them, there's 3 to 5 genders at least that we've known of. And so that means that their gender roles, if you want to call it that, also kind of ran the gamut in their culture. So you could see how it's all nothing. If that's like really recent too, it's, you can see how it all might be something that's kind of being imposed on what you found.
Yeah, it's an interpretation of findings, of remains based on current cultural attitudes. And I mean, like the finding of the Viking women warriors that Justin talked about a while back. It's like people hadn't really thought that Viking women were necessarily warriors too. But look at this. Maybe they were warriors, fierce warriors as well.
Yeah. So, but it's also one of those things that you have to remember about, especially when you're talking about a warrior class of the ancient world. You didn't need to be like the equivalent of a professional athlete today. You just needed to be well nourished, and you were that much more dominant than most people you ran into.
Right. Yeah, I think yeah, and this just goes into the conversation of modern ideas of gender, and so much of what we think of as just like, okay, we've got man and woman, man works, woman doesn't, woman stays home. This is because of a very it's using people, women, as property, and having it be a dominance through property and ownership attitude, as opposed to egalitarianism, everybody working together and trying to, needing to hunt for the village or the tribe or what, there are many reasons that we have embarked on our current attitudes, which thankfully are shifting. Let's talk about the darkness and how we can get away from the darkness. Do I just bring my platypus, Claire?
Yes, in fact, you should. So I don't know if you two remember, but I reported a while back on biofluorescence in squirrels. You remember this? The pink squirrels, the pink glowing squirrels. So the same team of researchers that was looking at these flying squirrels and how they glowed in the dark. They were going through all the specimens, the drawers of specimens in kind of the library and the taxidermy library. And while they were doing some of that, they actually decided, hey, let's shoot this UV light on this tray of platypuses that's right next to all these squirrels we're looking at. Whoa, what is that? Are they green? So it turns out they were green or cyan, which I'm assuming that's a kind of green. Anyway, so they were green under the UV light.
Cyan is like blue.
Blair,
oh, it is?
It's fine.
Oh.
I'm not colorblind. I have no idea what cyan is.
Okay, great.
I would have guessed it was like a pinkish red rust color. I have no idea.
Well, greenish or bluish, it sounds like. And so they found that these guys were glowing in the dark, which at this point, the only mammals that have been found to biofluoresce are the squirrel that I mentioned and opossums. And now if you're keeping track, we have the 3 different kinds of development in mammals, the 3 different kind of evolutionary radiation, if you want to consider it that way, of mammals. You have the monotremes, which are these first guys that lay eggs, and that's so platypuses, we have some biofluorescence in them. Then we have our opossum, which is our marsupial. So they give birth to very underdeveloped offspring and they go in a pouch. And then we have a squirrel, which is a placental mammal, just like us. So this is something now that has been discovered in 3 mammals, and they happen to be the 3 very different temporally, like developmentally in the evolutionary chain when they broke off, but also developmentally. So now this becomes a question of, first of all, I think we have to shoot UV lights on everything. No,
everything. Yes.
Every mammal.
Do you glow
in a museum? Get out your UV light and start taking notes, because either this is extremely widespread and it started in the very first mammals, or it is so helpful that it keeps popping up as convergent evolution, which, there's a whole bunch of questions about why they might want to glow in the dark. The best guess is that they are crepuscular, so they're awake at dawn and dusk. And so are the other animals that have been discovered to biofluoresce, so it might be helpful. But also remember that platypuses hang out in the mud. They move their beak around into the mud. They stir up all this mud into the water column underwater so they can get to bugs and stuff. So maybe it's helpful to be able to see each other through very cloudy water if you glow in the dark. I don't know. But anyway, this also means that they probably can see different wavelengths than we can as well.
Yeah.
So host of questions here. But ultimately, either most mammals can glow, or it just keeps popping up.
Well, I love the idea that they would be more visible during dawn and dusk. Where there's a little bit of light peeking in, and maybe that makes others of their kind. They're like, oh, there you are. I can see you a little better. I don't know.
Didn't we, Kiki? Maybe you brought it. I didn't. One of you did. That humans slightly bioluminesce.
That there
like
is
ever
work
so slightly, but
yeah. Yep, that we have a glow as well. We
got a little glow to us.
I mean, what if just everything does is the thing? Like, it's a wavelength. It's a spectrum of light. Like, just maybe we just all have some of it.
But then what's interesting is what can see in that wavelength. And what on earth is seeing this other reality of all the different creatures and how they glow? Is it bees? I don't know what does.
Well, that's also Thunderbeaver again. Just asked in the chat, would you want to see UV light? And 100% yes. I had a blacklight in my room in middle school and high school because I thought it was so fun.
Of course you did.
Yeah, but also it's more bright, which is closer to probably what the average person sees anyway. It's fine.
It's amazing. It's amazing. One more
platypus.
Yeah, I know. One more thing for this weird animal. Just add it to the bucket that stuff was thrown into at the time when evolution was taking place for the platypus. It was like, shake it all up. Oh, look what we've got here. Very odd monotreme animal.
So fun.
I didn't mean to make fun of you, Blair, for having a black light.
Oh no, you're spot. You probably could have guessed it, and you were right. I had a black light. I had a beaded curtain. I had a lava lamp.
Can I tell you? I had one of those black lights. I didn't have any
of these things. I had none.
I had a black light. I put it in my room as a teenager, and I had to get rid of it almost immediately because I also had really bad dandruff.
Oh no!
I was like, "Oh, this is gross. I don't want to see the dandruff on all of everything. Oh, this is terrible."
Well, yeah, that's awful. What if you wanted to look at your earwax? Do you have lots of earwax?
You check the Q-tip when you're done, right? Just to see
of course.
what kind of job you've done.
Yeah. Clean as a whistle.
All right. Well, there is a new methodology where researchers, doctors could easily check the level of your stress by checking your earwax.
Really?
Yes. Researchers from University of College London and King's College in London have published in the journal Helion that about a device that they are using that can be used at home without medical supervision to allow stress checkups and maintain social distancing. It uses cortisol levels in earwax, which the researchers say appear to be stable. Your earwax cortisol levels are there. It's a good, cheap, easy, effective way to get stress readings from a person. Yeah. So normally we can use saliva, we can use hair samples, but hair samples are much more long-term. They're time-consuming, and it's expensive to analyze them. It's cheaper to analyze with earwax. The researcher was actually inspired by honeycomb from bees, and because of that, went looking at earwax, looking to see whether or not it would make a good home sampling source that wouldn't risk a lot of contamination, knowing that beeswax is well preserved and can resist contamination itself. So they have a device that's like a cotton swab, and you stick it in your ear, but it has a break so that you can't stick it in too far and hurt yourself. It's got a tip covered with a sponge, and it also has a solution that is reliable for the samples. So anyway, they found that earwax samples yielded more cortisol than hair samples, and it was the fastest and cheapest method. Earwax for stress determination for the win.
But what if you're so stressed that you're constantly using a Q-tip and then you have no earwax for them to test?
Then you might have an issue.
So this is a reason to not keep clean ears, right? You want to let the earwax build up so that you can test to see if you're stressed.
Let your earwax build.
Yeah.
Test your stress. Yes, they are working on bringing this device to market so that it could be used as a medical device at home. Yeah, very interesting. Who knew? Cortisol in earwax. What you got next, Justin? Rats? Why do you have rats?
So the Palmyra Atoll, if you're not familiar, is a remote network of some islands out in the Pacific. Once upon a time, it had a rat problem. Now, rats weren't native to this island atoll in the middle of the Pacific Ocean. They likely arrived there as stowaways with the U.S. Navy all the way back in World War II. So they've been 60-ish years. They were there on this island with no natural predators. They basically took over the island. They ate seabird eggs, the native crabs, whatever seeds and seedlings they could find, and that was just Rat Island from there on out. So back in 2011, the U.S. Fish and Wildlife Service conducted a rat eradication project. They solved the problem. They got rid of the rats. UC Santa Barbara ecologist Hillary Young and her research group had already been tracking at that time another non-native species, the coconut palm. And they were sort of watching the coconut palm as it slowly began to invade the areas. It's considered invasive because it's not from that island, but it's also impacts nesting seabird populations because it changes soil composition, determining what trees can grow there. And the birds, I guess, don't like these coconut palms to hang out in. So they had plots there, and they were monitoring trees in various stages of growth and survival and throughout this atoll. This is quotey voice of Anna Miller-Turcule, I'm guessing, graduate student, researcher, and lead author of the study that appears in the journal Biotropica. Prior to the eradication, most of the understory, I guess that's the ground level, of Palmyra was either bare ground, sandy soil, or coral rubble, or covered in carpet of ferns. There wasn't a whole lot of trees. It was sort of barren there. Turns out the rats were quick to eat seeds and young plants coming out of the ground. They frequented the canopy as well, often nesting in the coconut palms and even eating coconuts. If you can picture this lovely tropical-looking island with palm trees filled with rats.
Yikes.
Okay, so rat eradication 2011. Did kind of work for a lot of things. The Asian tiger mosquito that had shown up on the island left again. Didn't have the rats to feed on. 2 species of land crab emerged that hadn't been there before, I guess, changing the atoll's biodiversity there. The only thing is, the foliage rebounded tremendously in coconut palm. Yeah. Turns out the rats were keeping down the coconut palm invasive species by eating all of their nuts, and so they couldn't grow, eating the little saplings as they would start, and eating the coconuts right out of the trees. They were basically keeping back the other pest. So,
time to pull out the palm trees.
Yeah, that's what they're doing.
Which one do you get rid of? Yeah, palm trees.
There's apparently millions of them now.
Yeah.
It's become sort of a problem. But this is sort of the interesting. This is the happy accident of the experiment is that they got to show an example of when you remove any species, even if it's an invasive species, if it's established, there are downstream effects that shift the ecology in other directions that you may not be anticipating. So,
I think that is the key point also, is the fact that biologists like to think they know the ecology of a habitat, of an area, all the things that are in it, but there are usually, not always, but usually unforeseen consequences. The ways that that organisms interact with each other. It's this it's like the genes and the markers in our genome related to height. There are over 10,000 markers related to height. How they interplay and how they interact with themselves and with other things in the genome. It's the same kind of thing. The ecosystems are complex.
Well, and it's just like any problem. You can't almost ever boil it down to one thing, right?
Exactly.
It's always there's always a multi-layered way to solve any problem.
Yeah.
And there is a tendency to tackle these problems as a one species problem. And, it's the Fish and Wildlife and also an island or nature conservatory, island conservatory groups that were involved in the eradication that identified the rat as a problem and what they can improve. They weren't obviously tracking the foliage, the plant, So even within them, they saw no other species that this was going to conflict with in the animal kingdom. And it looks like they're right. The mosquitoes weren't native. They're gone. These other crabs move in. All of that worked. They're like, aha, But yeah, it's slightly outside of your discipline, just one adjacent. You got to look there too.
Yeah. It reminds me of our conversation a couple of years back with Jess Phoenix, how she was talking about her project Blueprint Earth and how they're working on trying to get a blueprint for all of the different of ecosystems. What are all the things that are there? So it's not just the wildlife biologists talking about the wildlife by themselves and not talking about the plants with the plant biologists, and then you have the crab biologists.
Yes. And that's the thing. It's like if this had been anticipated, if this could have gotten put together and anticipated, you could have pulled out many thousands of trees before you got rid of the rats.
Mm-hmm. Yeah.
Instead of now millions and having the thing really. So it's. Knowing that the things are going to walk hand in hand allows you to be more proactive and use your resources better if you're trying to do a conservation effort. Something
that has been conserving its batteries, has a big conservation effort, is Voyager 2. The Voyager 2 spacecraft, which has been traveling away from Earth for more than 43 years, is now 125 astronomical units away from Earth. Astronomical unit is the distance from the Earth to the sun. It's 125 times the distance that we are from the sun right now. It is outside of our solar system. It's moving out of our solar system into interstellar space. It's out there. It's been doing that. And it was quiet for a very long time. It's very quiet. And it just called home. Yes. So last week, to test new hardware that was recently installed in the Deep Space Network, Voyager mission managers were able to send a series of signals to the spacecraft for the first time since March. And Voyager 2 called home and said, "Yeah, Mom, yeah, Dad, I got your message. I'm out here. I'm just fine. Doing my year abroad." Getting out there. Yeah. So Voyager 2 is still doing well, venturing out there beyond the solar system, investigating the great beyond. And it's just amazing that we have Voyager 1, Voyager 2, and now the New Horizons mission that are really out there, going out into the outer reaches of the solar system, beyond our solar system, and exploring. There's some new stories this week about thousands of Earth-like planets around, or at least potentially habitable planets around Earth, around stars like our own. And so if we could only get there, but we can imagine. And we can send our robots.
So Voyager's going out there. We just found out we can hear back. So that means that we can now grab some data from...
Yes. And we have been getting data from it on occasion, but because they were updating things and doing some new dishes and software and stuff, it's been a while since March that we've heard from Voyager 2. And, in the times when Voyager is offline and not responding to our signals, it's kind of, it's just recording stuff,
like
but it's got its batteries, its little nuclear power that it's moving on, and it's heading out there.
So this Voyager 2 is gonna keep sending us stuff, but at some point it'll be too far away.
Maybe. Well, it'll just take a long time for information to get back. Or there may actually you're right. At a certain point, there is decay. If the signal is not a strong enough signal to begin with, then you have signal decay over time and space, but it's going to be a long time.
I'm not great about how many astronomical units, because I think it's 90 million miles to the sun, something like that. That's like 10 billion miles away. It seems like it's
got some range on it.
Yeah.
Yeah. Hopefully we can get it out.
That's back when we had a thing called
cloud.
Radio Shack, and you could get NASA went down and just got these little signal boosters. It used to be a thing. It was really easy.
I mean, that's the technology that was used in the 60s and 70s. We think of it as very basic, but compared to modern technology and the strength of the computers in our handheld phones. But what's really amazing to me is that because of the small and delicate nature of technology today, it's more destructible. So interstellar solar radiation can damage the technology that we use in the transistors in the silicon chips that we use today. So you have to use more shielding. They have to be more protected. And those old spacecraft that we sent up, they were Radio Shack. It's like, put them together, let's get them out there, and it's going to keep going. It's just solid parts. They're going to work. Just like this show, which just keeps going and going, episode after episode, year after year. Oh, hey, if you want to help us keep going, head over to TWIS.org and click on the Zazzle link. I mean, buying stuff in our Zazzle store will allow you to get some cool stuff, but it's also going to support our show and will help us keep going and do the things that we do. Also, while you're there at TWIS, don't forget we have our calendars for sale. Dun, dun, dun. Blair did a good one. Yeah. Okay, should we talk about COVID now? Is it time? Let's have that.
Please, something relaxing to talk about. Yeah. Finally.
Can we? Because I don't know if I've been talking about anything but COVID for the last 8 months. Dear. Oh, dear. All right. There was some good news several months back about antibodies in llamas. Did you hear these stories about llamas having antibodies that could neutralize the SARS-CoV-2 virus? Perfect little antibodies. Well, llamas they have these little antibodies that are called single domain nanobodies, single domain antibodies, otherwise known as nanobodies. Nanobodies are found in camels and llamas, and apparently they had some good ones against SARS-CoV-2. Great. Well, we can't go around harvesting these nanobodies from llamas because, number one, there aren't enough llamas and camels on the world. It's expensive. It's difficult to raise all the animals for all of the antibody that would be needed to neutralize infections in the entire global population.
But what if I can get my hands on a llama?
You got to get your hands on a llama.
How much is that worth if I can get my hands on one?
Oh, everybody, if you've got a llama, lick your llama today. I mean, like your llama. Never mind. Don't listen to me. But listen to me about this science. So researchers have been trying to figure out how to get the antibodies, the nanobodies, without using llamas, without using camels. And so they've been searching for synthetic versions of these nanobodies, which are called Cy-bodies. Nanobodies to Cy-bodies. And Cy-bodies could be potential alternatives. Well, a group of researchers at EMBL Hamburg have used a platform of Cy-bodies to find some Cy-bodies that work to neutralize the SARS-CoV-2 spike protein. They had six of their analyzed Cy-bodies, SB12, SB23, SB42, SB76, SB95, and SB100, bound the spike protein with fairly high affinities, and these affinities are pretty good. So the Cy-bodies would be less expensive, easier to create than getting them from llamas. And the scientists performed a neutralization assay using a bunch of particles that were modified to carry the spike protein on the surface. And they say that 36 Cy-bodies were screened for neutralization, identifying 11 that are capable of neutralizing SARS-CoV-2 at a fairly high rate. So they found one in particular, SB23. It's a very potent neutralizer. And we'll see where this goes. Not the llama bodies. It's a Cy-body. All right. Beyond sci bodies. Oh, poor minks. Justin, you made it. You made it out of Denmark, but the minks aren't going to.
No, but
poor minks. We've talked before about the other animals that have a high probability of succumbing to COVID-19 infection. Mink, humans, cats, minks are among them and can catch COVID-19 at a very high rate. And in Denmark now, over 1 million mink have been culled in the country as a concern for the mink, and also that the mink will transmit the virus back to humans and could potentially be a sink where mutations in the virus could take place.
Which
Big
I
concern.
believe has already happened, hasn't it? I mean, I believe that there was an outbreak centered around mink farmers.
Yes, there has been.
Okay.
Yes.
So yeah, it goes into the mink. Then there's the other mutation. Then when it gets back into the humans, you might be dealing with a slightly different SARS-CoV than you had to begin with.
Right.
That's not cool.
I mean, maybe it would be the less transmissible version. I don't know.
That would be great, but
less bad for you version. The mutations
another
go
version?
all directions.
Do you want to watch that movie again? I don't.
No, let's not do this again.
No.
Yeah, but Denmark is among the countries that has definitely put a call from the government down on the mink farms to kill their animals because of concerns for infection, which is bad news for minks. But tell me about this leukemia super spreader, Justin. What's going on there?
Okay. So this is a study that details a leukemia patient who was actively shedding COVID for 70 days.
So that's a really long time to be actively shedding the virus.
Yeah, it's a really interesting. It's sort of a really interesting case. I mean, not completely unique, but very interesting. This is in the journal Cell. It's an unusual case of a woman with leukemia and a very low antibody count who was infected with coronavirus for 105 days at least. And was actively shedding that virus for at least 70 of those days, all the while remaining asymptomatic the entire time, despite being 71 years old, something along that lines. So this woman is, I believe, from Washington, and was in a nursing home that was heavily hit. One of the first things we heard about in COVID was nursing homes in the Seattle area. She's one of those. So she had gone to the hospital despite having no symptoms from that for some other reason. It was anemia, which is another thing that she had. So she has immunocompromised with chronic lymphatic leukemia. She goes to the hospital. They go ahead and test her because of where she was. Sure enough, she's got the virus. And so the first physician attending wanted to know if she was still shedding this virus, if it was still. So they got other people involved. Sure enough, she has. Munster, who's one of the authors of this study and one of the ones who's been tracking her, Munster says that as far as he knows, this is the longest case of anyone being actively infected with SARS-CoV-2 while remaining asymptomatic. We've seen similar cases with influenza and Middle East respiratory syndrome, which is also caused by a coronavirus. We expect to see more reports like ours coming out in the future. And she was getting the PCR tests throughout this entire period. So it was, yeah. Apparently they think maybe because she's immunocompromised, her body never made any antibodies, but it also didn't have the
inflammatory response. It didn't have the inflammation that is the problem.
Exactly. It's that second,
like I can't. I've had enough.
Yeah.
Yeah, resistance is futile. Yeah, I won't resist. Oh, well, we'll keep looking.
You just get in there. That's fine.
I'm just maxed out. I can't take another thing. I'm sorry.
But see, now, when we talk about testing, here's a fantastic example of why absolutely you'd want to test. Because there could be asymptomatic, meaning not coughing, not sneezing, no fever, none of the issues that have been reported as associated with the illness, but actively shedding for 2.5 months. How many people can there be who fall into this category, who are super spreading everywhere they go, but would never know it because they never got sick?
Mm-hmm. Right. Yeah. Testing would be so important. And there are probably many more who have compromised immune systems in one way or another. And Yep.
Well, and this is also why, right, the even though they say if you want to be safe, you quarantine for 2 weeks after you might have been exposed or whatever. But when we talk about if you did a real shutdown, it would be more like 6 weeks, is because you have to account for the fact that people spread longer than 2 weeks in outliers.
Mm-hmm.
Yeah. Final COVID story for this week. Results published in the Lancet's eBiomedicine journal from the study of the lungs of 41 people who died from COVID-19 in Italy found that their lungs were messed up. There was the normal lung structure was distorted. There was scarring of tissue, and there was a lot of blood clotting in the arteries and the veins. And the blood clotting of all of these together, but the blood clots especially, suggest a reason as to why the people we call long haulers take so long to recover from fatigue, from shortness of breath, and end up at a place where it takes months for the lungs to recover and the blood clots to clear for normal lung function to happen again. So this was a study out of the University of Trieste and King's College London, looking at these lungs from February to April. All 41 had extensive lung damage. 36 of the 41 had massive abnormal clotting in the lung arteries and veins. And 90%-ish of the lungs had a fusion of smaller cells into giant single cells with lots of nuclei, like the cells fused together. This is caused by COVID-19's spike protein, this fusion into what's called a syncytia. And
I didn't know that was a thing. Wow.
Yeah, it changes the cell membrane structure, stimulates the fusion of these cells with infected cells with normal lung cells, and that can lead to the structural changes. There's then inflammation and this blood clotting that we've been talking about. So this is a really interesting study, actually digging into kind of what is happening in the lungs and potentially pointing to an explanation for long haul effects, which we still really don't understand yet. But one step at a time. It's not pretty. And you know what? It hasn't magically disappeared yet. And we'll keep talking about it as long as it's here and as long as we're learning about it. Just letting you know that's a reality check right now, everybody. Additionally, there are many of our listeners are in the UK, and I know that the UK is potentially going into a lockdown because of their COVID situation at the moment. So I would love to know how you and our audience feel about lockdowns. What do you think? I'd love to hear from you and to get an idea about the variety of thoughts that are out there about lockdowns. So if you could write to me or send a TWIS message, or should I do a Twitter poll? Maybe I'll do a Twitter poll.
Yeah.
I'll do a Twitter poll in the next 5 minutes. And if you can all respond to my Twitter poll, it'll be out there. And I'd love to hear from you. And if you're in the chat room right now, you can let us know how you feel about lockdown and whether or not you think it's a good idea. Because we have talked about it a lot. Hey, this is This Week in Science. And if you want to help us grow, maybe, just maybe, you could tell a friend about us today. That would be really cool. Share us with your friends. just like your favorite pair of socks. No, that's not. Nope, that's not right.
Wait,
Just
what?
like your.
No, I'm not. My favorite socks. No, maybe my second favorite or third favorite I might consider, but not my very favorite.
No, share us with your friends just like a 6-pack. There's enough twist to go around.
From 6 feet apart, though.
From 6 feet apart. Yeah. Gosh darn it. I'm just not going. Okay. Do you know what time it is?
What's
that time again?
It's a certain time of the day when we like to sing a song and talk about animals. It's Blair's Animal Corner.
With Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. You want to hear about animals? She's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
So we talked earlier about brightly colored biofluorescing animals. I want to talk about drab, dull colored animals. We're talking about
Because
birds.
those are the ones that you can see. No,
Yeah, I
wait.
mean, I see it the same way you see it, which is nice.
Wait a second.
When we think about
Hand-colored birds?
yeah, when you think about birds, you're usually thinking about like
Real
parrots and birds of paradise and zebra finches and all sorts of fun colored birds. But there are some birds that are kind of drab looking, just kind of dull colored. And it turns out that the way that they see colors might be different too. So this is out of Duke University, and they tested the ability of different birds to categorize colors and found that it is not universal across birds. They selected 8 colors, varying continuously from orange to red. So this is already something I would have failed. And they made little discs with either one solid color or a combination of 2 colors. Birds were then trained to flip over the discs to show the 2 colored side in exchange for food. So set it no matter which side it landed on, kind of randomly, they have to change it so it has the half and half showing. But the idea is the closer those 2 hues get to each other, the harder it is to tell which side is solid and which side has the 2 colors. So this is how the researchers were able to look at what is called categorical perception. So there's a continuous variation, but we bin it into separate categories. So I immediately started thinking about when I do the calendar, for example, I have colored pencils that I use this year, and I have a 80-piece set of colored pencils, and I arrange them in Roy G. Biv, basically in a rainbow. But if you look at them, you would still call it purple, blue, and I'm just naming colors in the wrong order now because that's how bad I am at colors. There's a red section, there's an orange section, there's a yellow section, right? But as you get to those borders between the 2, like between orange and yellow, there's some that are like not quite orange, not quite yellow, right? But you still, in your brain, if you saw that separately, you would call that orange or yellow. You'd categorize it. And so, in this study, the female zebra finches, who they started with, presented with a continuum of orange to red colors. They categorized them in 2 distinct groups. Basically, they saw red, this is red, and this is orange. And this would track with their ability to see something that is relevant to them because they have orange and red beaks. And so that would kind of correlate to their need in the wild. So this is a signal of health where females are choosing their mate, how brightly colored their beak is. So then they looked at Bengalese finches. They are brown, black, and white, and they don't rely on colors when choosing a mate. They actually rely on song when they're choosing mate. So when they were presented with the same continuum of colors, they showed no evidence of a threshold between orange and red. This side is this stuff is orange over here, this section, and that section over there is red. Instead, they only picked discs that were on opposite sides of the continuum and totally different in hue. So basically they were me, right?
Yeah, they were like colorblind.
Yeah. So they paid more attention to the differences in brightness than the zebra finches. So they were looking at brightness, but in terms of the zebra finches, they were just looking at brightness as a way to reinforce differences in hue for the zebra finches.
Right.
So the way that these two different birds see color, the way that they see color seems to be very different, or at least how they categorize color in their brain. So as far as they can tell, their eyes and their brains physiologically are not any different. So it has to be about what's going on internally in their brain, right? Exactly. Yeah. So it has to be somehow behavior related. One of the things that they suggest is that Bengalese finches and zebra finches are both domesticated, but Bengalese finches are more distant from wild finches in terms of how they were bred to look the way that they look. And so it's possible that in the domestication process, they have kind of lost the ability to perceive because it became less and less relevant. So there's one theory that it has to do with domestication, but the other theory is just that it's the whole nature versus nurture, it's both thing. So they have a lot of the same equipment to be able to see colors, but it's not relevant to them as they grow up. So they brought this story because I actually think it's really relevant to what happened to me when I got my color blindness sunglasses. So the way it works is when you get them, you have to wear them for hours the first time you wear them before colors get to their full intensity. And it's because you're using part of your brain you've never used before.
Right.
It's not your eyes that need adjusting. It's your
brain.
So the sunglasses fixed the physical problem. And then your brain has to adjust, has to like gain plasticity to acknowledge new data that it's never had before. And so I think this seemed very similar to that. They might have the ability to see different hues, but it's unnecessary for them. And so they've never really put that much effort or thought into it, right? Of course, I'm anthropomorphizing now, but basically it was not part of evolutionary success to be able to tell hues of color. So they don't have as much, mental space, mental energy, practice, any of these things that help them to categorize colors effectively. So that's kind of the giant leap from this small study of two species of birds.
Right.
So again, this is a lot of speculation based on preliminary study. There's a lot more that needs to happen. They need to see if they can see something similar in wild birds or if they can expand it to more species of domesticated birds. We know there's a lot of finches out there. So you can just start pulling finch species and seeing how they can do with colors and if they have color signals in their mating process. And you could kind of try to draw these correlations and go from there.
Regardless of the mating aspect, there's also the side of it that food is colored. It's not drab, and being able to see color and recognize color and respond to color in the environment that's an important thing to be able to do. So I think
that's where this domestication thing could come in.
Yeah.
So maybe it's compound,
there's little
right?
Colored beads and seeds in that store-bought bird food that you feed to finches, the finch mix.
But I guess evolutionarily speaking, are you going to die if you can't tell them apart? No. It's all edible.
Yep.
There's no predators. You just get fed and you shovel it into your mouth. That's it. So it seems like evolutionarily, it might not matter to be able to see colors if you're domestic species, right? But so again, this would be a good reason to look at domestics versus wild birds and see if you see similar things. But the headline here is that the more dull colored species was not as good at telling colors apart, which, it seems like a pretty funny, weird, far out headline, but the implications when you dig down into it are pretty interesting.
They are
they better at
interesting.
seeing muted variations in color? That's what I would want to know. Are they just...
Yeah, that's a good question. Yeah, if they did
the same.
If they did the same thing with, or if they did the same thing, but with different types of patterns, right? Like, are they better at catching up on other visual cues? Yeah, great question.
I find it very interesting because then there's like the flamboyant cuttlefish with all those colors and its amazing eyes. So, the flamboyant cuttlefish is like, let me see everything. I have multiple color receptors and
right. But then, like, if given enough time or domestication processes or whatever,
would the flamboyant cuttlefish become drab cuttlefish?
Yeah. Or would they stop recognizing flamboyance? Heaven forbid.
Never.
Well, speaking of flamboyance, how about a fish surfing on a whale underwater?
What?
Yes, tell me more.
Yes. So this is a study from a team of an international team of researchers. They set out to study the fluid environments of blue whales. So originally they wanted to see, they wanted to do fluid dynamic analysis of whales. They could see what could be used as a basis to better understand behavior, energy use, ecological health of the species, and ways to improve tagging and tracking of whales and other migratory animals for future studies. But what instead they found was surfing underwater fish. So this is from, underwater fish as opposed to above water fish. Oh boy.
Right,
Remora fish. They're also called sucker fish. It's because they have a sucker powered suction disc on their head, and they are known to stick to the body of sharks or other large marine life like whales. And in the researchers studying the fluid environments of blue whales, this is off the coast of San Diego, they were able to capture the first ever continuous recording of remora behavior on a host organism. And they did that with biosensing tags with video recording capabilities. So they were able to have these kind of long-term data points of not just visuals on these surfing fish, but also the fluid dynamic stuff that I talked about before comes into play. Because how, as a fish, do you hang on to a whale as it's swimming, which is pretty fast,
very carefully,
and very carefully, and they move around while it's happening? So kind of like the surfer who's switching around his position, his or her position, and like going up on their hands and stuff. Well, in this case, the remoras move around, they feed, they socialize, they move all around that whale as it's swimming. How the heck are they doing that without getting flown off into the ocean? Well, they have some really intense fluid dynamics working in their favor as they are sucked onto the whale. So the whales sometimes go more than 5 meters per second, which is pretty fast. And so it all comes down to the drag on the whale. First of all, they select the best spots on the whale, which if you're a remora, the best spots to hang 10 or hang fin,
20,
I guess.
fin,
I don't know.
Right.
Right behind the blowhole. And then also behind the pectoral fin and behind the dorsal fin. And so these are the areas that I guess because there's, the fins make sense because they're reducing drag right in front. So
Yep.
They're catching all the water that's moving. And then the blowhole, there's in and out there's out movement. So I guess that's also going to create, plus I think there's a little cusp on the blowhole. So that's all going to mess with the fluid dynamics around that area. So it reduces drag. So the drag is actually pretty low. So if they hover off of the whale for a second, they're not just going to get pulled away in the current. But they also can move around to those different positions out on the whale. So you can see, those of you watching the video that Kiki's playing right now, they can move all over that whale at about 1 centimeter above the whale's skin. And so the way they do that is, I'm going to say a whole bunch of sciencey things now. Bear with me.
Go science.
Okay. In this narrow space between the remora and the whale, when fluid is funneled into a narrow space, it moves at higher velocity but has lower pressure. So it is not going to push the remora away. It actually sucks it back towards the host. So they can swim up into the free stream to grab a bite of food and then come right back down to the boundary layer. And it takes more energy to swim into the free stream flow than it does to just move around where the whale is. This is called the Venturi effect.
Yeah. This is
fluid dynamics.
Fluid dynamics used by boats and planes and all sorts of things. Yes, this is, and now we know by remoras, and it's basically once the remora is a passenger, it stays a passenger because it's just the best place to be.
Absolutely. And so now researchers can take this knowledge and use it to adjust how they tag whales, because when they do tag whales and other large aquatic animals, they often have all sorts of problems. Either they're too bulky, they might impact their behavior, they fall off in a couple of days, and that's a huge amount of money and time and effort to catch that animal, to tag it, and then it's gone after 2 days. So if they can use this information to make a new kind of tag, that would be a great way to collect long-term data. And also it just, it answers a lot of questions about the remoras. They've only ever had pictures, still images, and anecdotal evidence of this behavior. So this is the first time it's been caught on tape.
Kevin Jones in the chat room asking for a friend. What happens to them when whales breach? Which, that might be question. That might be, especially they breach, but then they kind of like slap the water pretty hard when they come down. Maybe that's the whale's way of saying, too many.
So I would wager a guess that the remora can probably feel the upward movement and would scoot over to the pectoral fin. So that a lot of time the pectoral fin does not breach. So they might be able to move their position on the whale so as to stay on when they breach. I don't know. It's when they're going fully out.
Question.
I don't know. They might not make it.
I don't know, but if you're just hanging on and you're a passenger, why not? I mean, fish can be a fish out of water.
Are they doing a symbiotic thing? Are they like cleaning the whale barnacles or something?
I bet the whales don't even know they're there. Although, actually, to answer that question about the cling, I do have a bit of actual numbers about that. So, the force of a remora's suction is about 11 to 17 newtons. And the force that a remora experiences when they are in the most intense place on the whale, underwater again, is 0.14 newtons. So they're hanging on pretty tight.
This, engineering overkill. That's the way you do it.
Yeah. Well, behind the blowhole, where they hang out the most, it's only 0.02 newtons. So definitely unnecessary. But maybe that's why. Maybe it's for the breach.
No, over-engineering. Maybe all about the breach.
Yeah.
Let's go into the breach with Patreon. If you would like to support This Week in Science, you can help us keep doing what we do by heading over to TWIS.org and clicking on that Patreon link and choosing your level of support. With your help, we can keep bringing a sane perspective, a sciencey perspective to the world that is kind of full of misinformation and disinformation right now. So be a part of helping us to combat that and bring science to the world. Thank you for your support on Patreon. Thank you for your support of TWIS. We really can't do this without you. We can't. Coming on back right now, it is time for Justin. What stories do you have?
So this story was sent in to TWIS by Minion Dave, who reminds everyone, time flies like an arrow, fruit flies like a banana. This is from the Max Planck Institute for Evolutionary Anthropology. Researchers have analyzed the genome of the oldest human fossil ever found in Mongolia, 34,000-year-old woman. who the DNA analysis show inherited about 25% of her DNA from Western Eurasians, demonstrating that once upon a time people could travel internationally. Ancient DNA extracted from the skull cap shows that it belonged to a female human who lived 34,000 years ago and was more related to Asians than Europeans, but had that connection still. Comparisons to the only other really early East Asian individual who was genetically studied to date as a 40,000-year-old individual who was found in a cave outside of Beijing, China, shows that those two individuals are related to each other, although the Beijing fossil does not have the Western Eurasian relationship in its ancestry. Interestingly, both individuals carry DNA from Denisovans, the extinct hominids that inhabited Asia before modern humans arrived there. Quotey voice, this is direct evidence that modern human communities in East Asia were already quite cosmopolitan earlier than 34,000 years ago, says Diendo Massolini, lead author of the study and a researcher at the Max Planck Institute for Evolutionary Anthropology. This rare specimen shows that migration and interactions among populations across Eurasia happened frequently already some 35,000 years ago. Researchers used a new method developed by the Max Planck Institute for Evolutionary Anthropology to find segments of DNA from extinct hominins and other genomes. They found the two genomes not only contain Denisovan DNA, but also DNA from Neanderthal, which is also interesting because Denisovan and Neanderthal are sort of themselves ancient cousins that split off long before current modern humans got to the area. But it also shows a path, perhaps, of out of Africa, mingling with Neanderthal, then keep going, mingle with Denisovan, keep going. So anyway, research is reported in the journal Science, and it is just continuing evidence that regardless of where you are on the planet, humans are a braided stream of evolution that isn't just the current modern human evolution braidedness either, but includes all sorts of archaic and other ancient human offshoots that cycle back into the genome that makes us us.
I like to be us. I'm glad there were lots of others before us to make us us. That's a cool find.
Yeah.
Very, very cool.
I'm acting as though I'm thinking heavily. I'm going into my next story, but it's taking a moment to load. According to the National Institute of Mental Health, there we go.
Good move.
According to the National Institute of Mental Health, more than 17 million people in the United States and 300 million people worldwide have experienced major depression. The rest of humans are just shallow and unthinking people. They just don't have feelings. Small study. This is a very small study of adults with major depression. Johns Hopkins medicine researchers report that 2 doses of the psychedelic substance psilocybin given with supportive psychotherapy produced rapid and large reductions in depressive symptoms. Most participants showing improvement, and half of study participants achieving remission through a 4-week follow-up. So remission in something like this is more than you expect from any form of therapy or treatment or medicine that is on the market, so to speak. Compound, which is found in so-called magic mushrooms, psilocybin can produce visual and auditory hallucinations, sometimes profound changes in consciousness and perspective over just a few hours of ingestion. Back in 2016, Johns Hopkins medicine researchers first reported that a treatment with psilocybin under psychologically supported conditions relieved anxiety and depression in people with life-threatening cancer diagnoses. Now the findings from a new study this is published in JAMA Psychiatry, suggests that psilocybin may be effective in a much wider population of patients who suffer from major depression than previously appreciated. Although it's just extrapolating from, cancer patient psychology shouldn't be that different than general public psychology when it comes to anxiety and depression. Small group, only 24 individuals. 67% showed a more than 50% reduction in their depression symptoms at the 1 week follow-up. 71% showed the reduction at the 4 week follow-up. Overall, 4 weeks post-treatment, 54% of patients were considered in remission, meaning they were no longer qualified to be called depressed.
That's amazing.
Really incredible. So
amazing.
This is one of those things that was initially used, and we've talked about this in the past, initially used in a psychiatric environment with amazing effects, then fell into the hands of counterculture and ran up against the government that was afraid of counterculture, and so was made illegal. It was technically just legalized in the state of Oregon.
In Oregon, where I'm located. Yeah, it's
as well as for research. It's been opened up there for research purposes.
Yeah, it's for medical treatment, so research purposes and for medical treatment for conditions like depression and anxiety. So
I think also.
It's not for recreational use.
Yeah, I think also mushrooms have been decriminalized in Washington, D.C., which...
In Oregon, we also decriminalized, I think,
everything.
All recreational drugs.
Everything.
This election, yeah.
I think it's interesting that the way medicine, pharmaceuticals, I guess I should say, has kind of moved in the modern era. Is like you have all these natural remedies that have been used historically by native cultures and all sorts of people. And then there was a push to distill down exactly what chemical it was or find a chemical compound and make it synthetically. So there's this huge kind of swing that way. And now we're developing kind of a little bit of a swing back where we're looking at the natural origins of things, and maybe that's not so bad, which I think is great. It's as a pendulum always works, we should really be in the middle. We should be looking at like the scientific implications of a thing, and then maybe not trying to mess with it too much, because now we know, like, maybe you find what you think is the thing in the mushroom that is the beneficial thing, but if you make it in a lab, maybe it won't work the same way as the mushroom did. So there's the
law has changed in Oregon. So the science is actually going to be able to be pursued after a 60-year hiatus from reason.
Hopefully it leads to research happening here in Oregon. Hopefully federal law does not come in and negatively impact this state decision. But we'll see. We'll see where it goes. Things that can help people are always a good thing. Let's talk about helping people, changing people's minds. Do you know how much corporations change our minds? So it's ridiculous. It's really, really ridiculous how much corporations are changing our minds, influencing our decisions, sometimes without us even knowing it. A researcher published a new study this week in the journal New Media and Society. Researcher Dr. Taneja is a professor of media at Illinois, and the co-author Wu is a professor of media culture and communication at NYU. Their study is called Going with the Flow, Nudging Attention Online. And in the title, flow that they reference is a concept that used to be applied to radio and television, the audience flow. And that was something that was defined by programmers who programmed the content for television and radio. Nowadays, we've got a la carte stuff, and you can hop from Netflix to Amazon Prime to, Hulu to YouTube to, wherever to get your content. But there used to be a very directed flow that would take people through the evening and keep people on their channels and watching their shows and seeing their advertisements and hopefully buying things and having certain perceptions and perspectives as a result of seeing what they watched there. And so they wanted to take that and apply it to the internet and to see whether or not there are predictable patterns of how people go from website to website and how that happens. And so they took a bunch of data from a data research company, ComScore, back in October 2015. And their sample was based on 1 million internet users, including 1,761 websites that reached at least 1% of the U.S. users during that month. And then because of all this data, they were able to identify clusters or constellations of websites that represented sequences that were common within behavioral patterns for users, for browsers. And it was all browsing behavior that was linked to corporate ownerships, partnerships, and website types. The researchers say that they identified 11 clusters and the anchor sites within these clusters that served as the common starting points for how people start their browsing and get going. And among the clusters are Bing Microsoft, which is anchored by Bing and MSN content sites. A Google cluster, of course, anchored by Google search. A lot of us use that, and the Chrome browser, YouTube and Gmail, and a social media cluster that's anchored by Facebook, Twitter, and LinkedIn. And then there were two other clusters, the Yahoo cluster and the AOL cluster, that they surprised the researchers because nobody thought they were going to still be relevant, but they think it's possibly because of older users using these clusters. There were...
Hey! I am part of the Yahoo cluster. I admit it.
Do you search with Yahoo?
No, I don't. Oh, okay. I don't search with it. No, I Google everything. I only Google. I don't even like results. When I get results from other browsers, I'm like, that's not how Google would give it to me.
That's not how I like it like that. Go away. There were some other clusters too. They were surrounding data solicitations, retailers that use Citibank as their banking backend, pornography sites, job search, and travel. These were various browsing clusters. And there were 4 methods that corporations nudged online users. And this is the word that they use throughout the study, is this nudging, that corporations are using technology, using the websites, they're using wording, they're using all back-end stuff to nudge people in certain directions online, and we're not always aware of all of this. Then these methods, they're at a different level of visibility and control. And so the highest was content ranking and curation. And so search engines and social media do that. You do the Google search, what's at the top of the algorithm. The search engines are basically ranking things and telling you where to go. Social media also have an algorithm that tells you what to look at based on what they think you should look at. Then there's hypertext. These are words that are linked within the text. And additionally, the nudge is visible, but I guess the users don't have as much control. Third type of nudge is employed by Microsoft through software configurations that are built into the Windows operating system itself, like making Bing the company browser, search engine, and homepage. all by default, and that when it's in there, people aren't necessarily going to change it, right? Whatever's on your computer, you're like, I just use what's here. And so that nudges people into certain browsing patterns. And then finally is the back-end databases and software. That's e-commerce, like Citibank, and there's credit card payment retailers. And then additionally, there's all sorts of stuff that's, on the marketing side of things where there are funnels, and people talk a lot about funnels that you get pushed down if you click certain links moving certain directions. So really, it's that people don't see the internet as a space that operates in this kind of behavior directing way. People think they're directing their own behavior on the web, but there are actually lots and lots of ways in which we are being nudged by the corporations in the world that own the pipelines, that own the web spaces to do what they want us to do.
Mm-hmm. Welcome to the machine. What did you dream?
Welcome
It's all right. We told you what to dream.
The machine. Yeah. Welcome to the machine, everybody. Oh yeah. And then in last kind of cool story that has nothing to do with nudging, but is awesome. There's a bunch of researchers who did a bunch of calculations on black holes and trying to figure out the black hole information paradox. There's like a bunch of papers that just came out, and they've been calculating and these.
I looked at it. I didn't understand it at all.
Yeah. I don't know that I understand it.
They made it sound very exciting.
Yes.
But I did not understand it.
Well, the question is, what happens to information when it goes into a black hole? And Hawking and others, they decided to use quantum theory to describe matter in and around black holes, and that gravity would also have to be described using quantum theory. And there's a hybrid approach. Like Hawking and his colleagues, they thought they figured it out, but basically they're wrong. They're not completely right. And through not really looking at it as only Einstein's classical or only quantum theory calculations, and actually
and Leonard Susskind helped illustrate all this too. Yes.
Yeah, it's basically over time they found that they found new gravitational configurations that Hawking didn't include. And they dominate, they get to be more prominent when a black hole gets really old. So black holes age, everything gets older. And as black holes age, apparently they don't want to hold on to information anymore. And it goes from being this quantum system where gravity is treated in a quantum fashion when they're young to it just being gravity and information going in and coming out. And they don't really know exactly how all that happens, but when black holes are really old, they're just like in one ear and out the other. They just do not want to hold on to anything. So they're just like spitting information out all over the place. But this is the new idea. It's this new calculation based on a hybrid combination of physics ideas. But yeah, it doesn't.
I need like an analogy with like a pool table, a rubber mat, and a swinging pendulum. Maybe, I don't know.
Maybe, yeah.
The worst one. The worst.
I just like thinking of really old black holes who are just like, I don't hear you, and they spit it out. Whatever.
The worst analogy was the one where it's like, okay, so you have a bathtub full of water, and you put a couple of drops of food coloring in, then you mix it up. How do you get that information out again? I don't. Why would you even try? I don't. Yeah, maybe this is something we should reach out to one of our physicist friends to try to
come and explain this.
Slow-splain me.
Mm-hmm. Yes. Well, maybe we can get Ethan back on. Ethan Siegel is pretty fantastic at that.
Absolutely.
Yeah. And then leading us out of the show, we do have a question for tonight. This is from Fada. Fada says,
"Hey, Fada.
Here's my head scratcher ponderer for your TWIS user questions section. This question either shows I'm incredibly brilliant for thinking of something no one else has thought to do, or I'm incredibly stupid and have no idea what I'm talking about or how astronomy actually works. Either way,
that's how I roll.
I think it's a fun thing to ponder.
Yes.
So when our best visual telescopes view phenomena in our galaxy and the universe, be they the Mount Wilson Observatory, the Cerro Paranal or Cerro Tololo observatories of Chile, the Hubble Space Telescope, or the future James Webb Space Telescope, we have to deal with the fact that due to the whole speed of light thing, what we're looking at is a snapshot of how whatever object we're looking at looked that many light years ago that it is distance from us. So when we're looking at like the Pillars of Creation nebula, we're not looking at how it looks now. We're looking at how it looked 7,000 years ago because it is 7,000 light years from Earth. And it took its light 7,000 years to reach us. When we look at the Andromeda galaxy, we're actually looking at what that galaxy looked like 2.5 million years ago. For all we know, any of these objects could have ceased to exist by now. So the question is, given our best guess as to where the universe's center is moving away and expanding from the Big Bang, and given that we could estimate the general path or arc of our Milky Way galaxy away from that original Big Bang and a universal expansion, would it be possible to get to a point to point a powerful visual telescope like the Hubble back along that path that our galaxy has traversed from the universal center and actually take an ancient picture of our own galaxy from thousands or millions of years ago? Like a galactic selfie? Where's this one going to fall best?
Right, we're supposed to answer that question?
Yeah, what do you think, Justin?
Yeah, so I think what we would have to do is travel at the speed of light. Maybe faster. And then go really far away and take a bit. So here's the thing with taking a selfie. We already have a nice selfie of the Milky Way. We should be satisfied with it. We should go ahead and use it as our profile. It's when we shoot towards the middle. We're out in the rurals. It's basically like right there, like Kiki did right there. Yeah, her selfie, her hand isn't in the picture. She takes a picture, right? She's missing her hand. She's missing the entire back side of her. Probably is only, shoulders up, ish. So really the picture you get even with a good selfie isn't that great. And so the just focusing on our Milky Way is basically the best selfie we're ever going to get, of Milky Way. It's our own center.
I feel like you'd have to go too far. Well, I guess if you had an infinitely,
you have to go ahead.
You'd have to have a telescope that's like way better than anything we have, right? Because I think it's all a matter of, if you're talking about light years,
telescopes are fine. You have to travel. You have to take the telescope many.
You have to take the
years.
telescope to wherever it is.
I think what he's asking is, if you
can you look back?
We're in orbit around Earth with a telescope.
Okay.
Could you point it at a direction where you could see the Milky Way before now?
No.
So even though you're currently in it.
No, we're in it, so we can't see it before it was here. We're not there now, though, so we can't capture that light.
Right. And that's the answer. Is that because we're not physically there now? That's really the key. Because we're not physically there now, there's nothing to capture. We would have had to take that picture then, and we just wouldn't get the picture until now.
Yeah. Yep.
Oh, the other way you could do it is you could invent a time machine that could travel in time, but not space, meaning it stays in the same point of space relative to where it was. You can travel 1,000,000 years into the past, but not move in space, and then look back and you could see where the Milky Way was 1,000,000 years ago.
Just in time. It's the end of the show. Yeah, we've made it to the end of our show. Thank you, everyone, for listening. And if you have a moment and you haven't bought a TWIS calendar yet, head over to twis.org and click on the froggy toad and buy a TWIS calendar for 2021. Blair has made arts for you. It's time now for the shout outs. I would love to shout out to people who help support the show. Shout outs to Fada. Shout outs to Identity 4. Shout outs to Gord. Thank you for all the things that you do for the show. And thank you to the Burroughs Welcome Fund and our Patreon sponsors for their generous support as well. Thank you to Donathan Styles, a.k.a. Don Stylo, John Scioli, Guillaume, John Lee, Ali Coffin, Maddie Perrin, Gaurav Sharma, Josiah Zahner, Mike Shoemaker, Sarah Forfar, Donald Mundus, Gerald Sorrell, Stephen Alberon, Daryl Myshak, Stu Pollock, Andrew Swanson, Fred S104, Corinne Benton, Sky Luke, Paul Ronovich, Ben Bignell, Kevin Reardon, Noodles, Jack Brian Carrington, Matt Bass, Joshua Fury, Sean and Nina Lamb, John McKee, Greg Riley, Mark Hessenflow, Gene Tellier, Steve Leisman, Ken Hayes, Howard Tan, Christopher Rappin, Richard, Brendan Minish, Melizond, Johnny Grinley, Flying Out, Richard Porter, Christopher Dreyer, Mark Massaros, Artyom, Greg Briggs, John Atwood, Robert, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Matt Suttermatt, Philip Shane, Kurt Larson, Craig Landon, Mountain Sloth, Jim Drapeau, Sarah Chavis, Alex Wilson, John Ratnaswamy, Sue Doster, Jason Olds, Dave Neighbor, Coast D. Ranke, Matthew Litwin, Eric Knapp, E.O., Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Stanton, Paul Disney, Patrick Pecoraro, Ben Rothig, Gary S., Ed Dyer, Tony Steele, Ulysses Adkins, Brian Condren, Jason Roberts, and Dave Friedel. Thank you for all of your support on Patreon. And if any of you listening right now are interested in supporting us on Patreon, you can find information at TWIS.org. On next week's show, we will be speaking with science writer Marta Zaraska about how to grow younger while growing older. Don't miss it.
Otherwise, you'll be the only old person, and the rest of us will just be younger.
Young. That's right.
But we will be back next Wednesday, 8 p.m. Pacific time, broadcasting live on our YouTube channels, as well as the Facebook channels and whatever other twist.org/live channels and whatever else is out there.
Hey, do you want to listen to us as a podcast? I know you'd rather check out all of our amazing hair today instead, but if you want to listen instead, you can just search for This Week in Science or where podcasts are found. If you enjoyed the show, you can also get your friends to subscribe as well.
For more information on anything you've heard here today, show notes and links to stories will be available on our website, www.twist.org, and you can also sign up for a newsletter.
You can also contact us directly. Email Kirsten at kirsten@thisweekinscience.com, Justin at twistminion@gmail.com, or me, Blair, at blairbaz@twist.org. Just be sure to put twist, TWIS, in that subject line or your email. It'll definitely get spam filtered into the sucker of a suckerfish who's surfing a whale.
Yeah, that's
where
back.
is actually an opportune place to hit us up on the Twitter. We are at Twist Science, at Dr. Kiki, at Jackson Fly, at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We will be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer and it shouldn't be news. That what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. 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 And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 Got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you've learned anything from the words that we've said, then please just remember it's all in your head. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science. This Week